So Cara Magnabosco is an assistant professor of geobiology at the ETH Zurich. She spent some time in Greg Fournier's lab at MIT and some time at Flatiron Institute in the Simons Foundations in NYC. Her research focuses on the development of computational methods for investigating the coevolution of life and the Earth, and particularly the expansion of metabolisms throughout Earth's history. She uses ideas derived from genomics and phylogenetics in order to detect signatures of ecological and environmental changes contained in the genomes of modern microorganisms. Cara?
Hi. Yes, thank you, Jana, for the kind introduction, and thank you to the ENIGMA team for the invitation to speak here today. I think it's been, as a geobiologist, it's been super exciting to hear all the different talks and unique perspectives about how to look at and study the sort of coevolution of life on Earth. And so when I was invited to talk, I guess I was kind of asked to give my perspective on this topic. And so I wanted to kind of describe the sort of statistical models that we use in order to approach this. And so I think you've kind of seen different flavors of the kind of things that I'm interested in and my group really works on, which is thinking about evolutionary inference and evolution through reconstructing phylogenetic trees. So here, again, you've seen a few different versions of phylogenetic trees. Here is just an illustration of one. Another one where you have bacteria in blue and archaea in green. And what you can see is at the tip would be organisms of genomes sampled today. And these paths going back is the evolutionary history of each organism back all the way through a path to the last common ancestor. And so this is one representation of the evolutionary history of life itself. And it's very, it has a lot of applications, including what I would call here some of the biogeological models or geobiological models that we use. So I'll talk a bit more about this throughout the talk, but this is just another representation of a phylogenetic tree. Again, you have some common ancestor going into different specific groups. And there's something unique about this tree. It's actually a time tree, something built as a molecular clock. And so what you have here is that the branch lengths actually correspond to some amount of time. So Jana introduced one time tree, and so we'll talk about specific time trees that I like to use, which are horizontally constrained, horizontal gene transfer constrained molecular clocks. And the reason you can take these another step further, which I would call sort of the geobiological inference problem, which is given a tree or having some model of how microorganisms evolve, you return some posterior distribution of the age of certain groups, and you can overlay that with questions you might have about certain signals you see in the rock record. So in this instance, I have overlaid the posterior age distributions of Cyanobacteria, stem and crown Cyanobacteria, with sort of just this schematic. There's many different ones, but here's just one from the Lyons paper, 2014, where you have very little oxygen, the great oxidation event, and the secondary rise in oxygen. And what you can see is that we can look and say, okay, what is the probability that crown Cyanobacteria were around before the GOE? And we can see based on this that it's very high. I think it's something like 98% based on this clock model. And stem Cyanobacteria, we're really certain that Cyanobacteria, this stem lineage was around prior to the great oxidation event based on the model of this paper, which I'll describe a bit more about how we do that. And then sort of the next step that we're really interested in thinking about is how to tie this all together. We're thinking about the evolution of populations, or evolution of lineages across time, and they're kind of just in this abstract space, but there's a lot of other things happening on the planet during the course of planetary evolution. And so I think this is a concept that has been sort of introduced and shared to me with me from largely from one of my colleagues at ETH, Tarsgaria, who's a geophysicist, is this idea of biogeodynamics. So linking the evolution of life with also the changing planet, in particular from the geophysicist perspective, something that's really interesting is this evolution of plate tectonics over time, where you're changing sort of the regime and from this, what they call squishy lid tectonics, and transitioning into what we actually view as modern plate tectonics today. And this has a lot of implications for thinking about different biomes, the size of biomes, as well as, you know, how strong different sources and sinks within our planet are throughout Earth history. So I think that's still a bit of a work in progress, but I think one thing I want to convey is, you know, there's this question about the coevolution of life on Earth is, you know, a very multidimensional problem. And so I think one thing is what exact models we use. And then the second part that I'll want to get into a little bit is how do we go and try to find additional features that can be helpful in understanding a bit more about the coevolution of life in our planet. Okay, and so before I get into some more specifics as to approaches and findings that we've had in this area, I just want to pose, you know, a success of another field, sort of the ecological paleobiology field, which I think is becoming also gaining a lot of progress in the tools that they're developing and the approach that they have, and perhaps be interesting to discuss later, you know, whether or not you think there are ways to kind of combine these ideas into the deep time problem. And so this is somewhat an older idea, which has been more recently shown to have some basis, which is the idea that as you have plate tectonics going from the supercontinent into the present day geographical formation, this breaking apart of Pangaea actually increases, leads to an increase in biodiversity. And there's a few different models that people are using to go back and kind of study this effect it has on different groups of animals, such as niche modeling, paleogeographic reconstruction. And they're really finding a way to meld the idea of a changing geosphere with the changing biosphere. And I guess one thing that is important to point out about these approaches and why we can't just go and directly use, you know, the ideas from paleobiology into deep time is that what they're depending a lot on is the fossil record. And not only the fossil record, but also a very good rock record to reconstruct climate or locations of the continents. And so with that, we're kind of left at the question is, what is that signal that we can look for from life and in the genomes or the biomolecules of life to help us get at, you know, much more ancient times where fossils are disappearing and the rock record is sparse. And so I think we heard some really interesting ideas from other people, and I'm really looking forward to chatting more about the ideas that were discussed as far as small folds and metabolic networks. But for this part of the talk, we'll really just focus on, you know, the more traditional idea, which is looking at DNA or the proteins that are encoded by the DNA and using that to reconstruct an evolutionary history of a microorganism. And so this is just the very simplified picture that you get from your high school biology textbook, that you'll have some DNA sequence. And because DNA replication isn't perfect, over time you'll accumulate mutations. And sometimes these mutations may lead to something called a frameshift mutation, which would actually lead to a change in your proteins themselves, which may also imply some difference in functions. But for now, we'll just think about these individual point mutations changing each time we replicate. And so the earliest models of molecular clocks were based on this idea that, you know, you're accumulating changes in your genome over time because of the imperfect replication. And you may be able to go on a phylogenetic tree, which is again the path of how the DNA sequences are changing and evolving into unique groups. And you could say, okay, if I see that this ancestor maybe has 5 mutations accumulated, and the rate and I see that there's 1 mutation every 10 years, then this was 50 years of time between the 2 groups. And of course, that's basically what is called a strict molecular clock, and we know that this has some issues. So a lot of work has been done to improve, you know, these molecular clock estimates. And what we use now are something called relaxed molecular clock models, where we can actually vary the rate across the tree to get more accurate estimates of ages for different ancestral nodes. And so I guess, and so the question is, okay, sure, we can draw rates from some sort of distribution and say, and use the sequences at the tips to give us some indication about how well that rate may fit our explanation of how long this divergence may have been, but without any internal constraints, say knowing that this node here was somewhere around 500 million years to 600 million years old, my model may return something like a 1 billion year range of in the posterior, the divergence age estimate. So you won't have very accurate age estimates if you have no internal constraints. And so the question is, it's kind of this chicken and egg, can we use these models for microbiology when, as I said earlier, we don't really have a very good fossil record? And would it be appropriate? Or the secondary, is there an alternative piece of information that we can use that can act as a constraint, and we can implement that into this existing molecular clock model that was developed for, you know, fossils, more fossil-based paleontology applications. And so this is some work that I started working on when I went to MIT for a year to work with Greg Cornier's group. The idea was that, you know, there are very many fossils that can accurately place a timestamp on different bacterial lineages. But within the genomes of bacteria, there are these scars, notably what I like to call scars, horizontal gene transfers, or instances where a genetic element was transferred from one organism to another, and then subsequently kept fixed into the population and subsequently passed down over time. An important thing that's happening within the horizontal gene transfer that we can leverage is that if there is an actual exchange of genetic material between 2 organisms or by a vector, these 2 organisms must be more or less contemporaneous to one another. And so what the hypothesis was when I started working with Greg was, you know, can we actually utilize horizontal gene transfers as a secondary piece of information to further calibrate molecular clocks? And so this is just here a picture of, again, the tree, and in red, it's real data. These are actual transfers happening in the trees. And so as you can see, there's even some really strong tendencies of certain groups to transfer specific genes to one another that are not closely related at all. And so this is the working hypothesis. Can we take some gene tree that we see? So here's just the illustration of what a gene tree with a horizontal gene transfer might look like, overlaying the species tree, where you have a gene history very much tied within this blue lineage, but then a transfer has happened here, And this subgroup here, this teal group, has also inherited the gene. And as I said, what I can tell from this tree is that, okay, this event they must have been somewhat contemporaneous. So I can say that this node here, before the transfer happened, is older than the last common ancestor of this node here. And I can use this type of information to go from my molecular clock, which gives me divergence state estimates for specific ancestral nodes, A, B, and C. Here they're just shown as the posterior distribution over time. I saw that there was this transfer between these two groups. So now I know that C is older than B. And so I can invoke this conditional constraint, which will readjust my tree and show that, you know, this is actually what the evolutionary history looks like, showing the proper ordering A is older than C, which is older than B. Okay, and so this kind of idea was the foundation of some work that we did, and we're now, what I've been working on a lot now is kind of making this more general so that we can apply many horizontal gene transfers at once during the actual inference problem. But I'll show here just more of the example case of that first picture I showed, which is, okay, when did the different phototropic groups evolve? In particular, let's try to say something about cyanobacteria and the relationship with the GOE. And so this is, you can refer to this paper here to get more specifics on the methods, but the basis of it is shown here. What we do is we construct a phylogenetic tree, again, and we include the groups of interest that we were really interested in dating, as well as, you know, their closest sister groups and some other groups that may have informative calibration points to be included in our tree. And we identify various fossil calibrations or specific geologic calibrations that we may be able to link to a certain group, such as the ancestor being, you know, an age range between the last zircons, or the early zircons, and then the stromatolites, as well as, say, the mitochondrial ancestor, right here in alpha proteobacteria, as well as by these dotted lines, these are the horizontal gene constraints that we apply, saying certain groups are older, like 6 is older than 8, I believe was the transfer, and another one here, which would be the relationship between 5 and 2. And what's really useful about these transfers is not only do they link groups that are very far apart, but they also link, you know, information that we have of fossil constraints to groups that are otherwise unconstrained. And when you do this approach, what was really exciting to see is that, yes, indeed, you do see a great improvement in the precision of your divergence state estimates. So here is just the divergence state estimate of green non-sulfur bacteria when you don't use a horizontal gene transfer constraint. And then this is if you do use that horizontal gene transfer constraint, you actually get a somewhat informative age range, whereas without it, we're getting about 200 billion years. And it would be very unclear to say anything about how green non-sulfur bacteria evolved in relation to the changing Earth. And so as I was saying in the beginning, it's not just a date estimate that we get saying this group is this old, has origins at this date, but it can also be used, we can use these models in another way to do hypothesis testing, such as is this age older than a certain event? Or across the tree, we can compare posterior distributions between groups to say, one thing we explored here was whether or not the duplication of photosystem 1 and photosystem two within the cyanobacterial stem group, if this could happen and then be subsequently transferred to green sulfur bacteria and green non-sulfur bacteria, is that a feasible scenario? And we can show with high certainty, yes. Whereas the alternative scenario of transfers from green non-sulfur bacteria and green sulfur bacteria into stem cyanobacteria is less likely to occur. And this is just some hypothesis testing that can be done with molecular clocks, which I think is very useful and I hope can be used in the future as a way to help evaluate, which scenarios are more or less plausible in the many scenarios that are often given for explanations as to certain geologic features that we see in the environment. And so I think I have maybe just I have just a few minutes left on the time. So I wanted to say that, this is one such model, and we're working to improve this model that we can make it more general and add additional constraints, make it computationally tractable. But then the next, the other side of the problem is, horizontal gene transfers. We see that there's great success, that we're getting a big improvement in our divergent state estimates by adding this piece of information. And then the next question is, what are additional pieces of information that can help improve our models even more? What features, what genetic features can we use in various inverse problems about coevolution of life on our planet? And so I think I'll just have a speed through this really fast, which is this idea of getting to biogeodynamics. And I just wanted to highlight an interesting analysis that you may be familiar with, which is this study from 2003, which was looking at the different Heliobacter pylori from a very large cohort at the time, 370 different isolates. And what they did was using, the admixture model, which is very common in genetics, to infer what are the ancestral strains of these isolates. They identified that there were three subpopulations that were the ancestral populations. One from East Asia, Central Asia, and Africa. And they could see that based on the distribution in present day, it seemed like that these microbes were actually being passed in a similar, were being transmitted in a pattern that mimicked human migration. And I think this is, we've been looking at another gut microbe that H. pylori is kind of unique because it's often thought of a pathogen. I guess there's some debate as to whether it's really a pathogen or more of a commensal, but we looked at a true commensal, and I think with a lot of other commensal gut microbes, they're seeing more and more a very tight tie between the evolutionary history and sort of the distribution of these organisms matching human migration. And so I guess the question is, are there similar patterns that these are things that are very tied to the host within their genomes? Of course, not all the sites within the human microbes biome microbes are predictive of human migration. Some are predictive of other features. And so which genomic units are good for, human migration, which are good for diet, which are good for other aspects. And then more maybe, in the larger scheme, thinking about organisms that are not associated to those, but just free living organisms, which make up the majority of the microbes on our planet. How are they evolving, and which specific sites or features on their genomes can be leveraged to elucidate information about the Earth history? And so with that, I'd like to thank, give shout out to my group, my growing group, and these 3 people who I've been working with, and hope we can make some more progress on these exciting topics, as well as just here's some of the references of the papers I was talking about, and just a shameless plug that My group is relatively new, and if this kind of work interests you, reach out, and yeah, we're hiring. So thank you.
Thank you, Cara. Thank you very much. I'm going to ask you a very simple question.
Yes.
So, what is the stem group of cyanobacteria?
So what is the stem? So if we go to this picture, I think it's easier with the picture. It's something that it doesn't exist anymore, so we wouldn't be able. Obviously. We don't know what this is. But if you look at this, so stem and crown are terms that are referred to within the tree. Yeah, sure. So the tree is the last common, the crown is the last common ancestor of a clade, which right here is, I guess it's easier with the actual tree. Okay, sorry, I don't know where it is. And then the stem is the last common ancestor of that clade to the sister, what we would call the sister group. And so the reason you get this, what we call this the distance between the stem and the crown, is that there are many groups that appear within here that are no longer alive. They went extinct at some point in the past.
So, but did the stem group generate oxygen?
Yes, I think that what we believe is that at some point there is some stem group member that was making oxygen based on this model.
So can we couple that with geochemistry? So
in what way? I think that's the question is okay, if we know that we can say that there was oxygenic photosynthesis at this time period, then the couple would be that oxygen, this term for oxygen production, we can have it be positive. And so that would be one possible coupling.
Right, I'm thinking of geochemical proxies, but that's not the issue. I think the issue is we have an uncertainty of the evolution of cyanobacteria, which is not the most, but is one of the most important groups of organisms on the planet, of about 1 billion years.
You mean based on here between the distance between the stem and the crown?
Not just on genomic distances, but or inferences, but on geochemical
on the proxies.
Yes.
Yeah.
So it's actually an embarrassment to me. So I can't imagine that you had a photosystem 1 and a photosystem 2 that could have evolved early. And they, we think in photosystem science that they came from one common ancestor. There was one reaction center that became the photosystem 2 type reaction center, and the other became the photosystem 1 type reaction center. That's one hypothesis that's very popular. Okay, this is a discussion of that's probably a bit too technical detail, but I'm just trying to understand when we got oxygenic photosynthesis.
Yeah, and so I think it's tricky just by looking at one gene or one piece of information. I don't know how much better we could get with just these pieces, but what I think will allow us to get more and more precise is by looking at what additional features might there be that can even tune this in even more. And so I think that's what's the real hope, because there's secondary effects that we should see embedded in the genetic history, perhaps the protein structure, protein fold, perhaps the metabolic networks, perhaps something that we can relate through ecology or population dynamics, and get a better handle on that uncertainty.
Okay, thank you so much.
Thank you.